Okay, so let's turn to the last segment, and that's what's coming. And fortunately, on this panel, we have key investigators on these clinical trials. So I want to jump right into it. But first, let's start with you, Jamile. I mean, why do we need new therapies? I mean, you know, why is it important to find these alternatives?
I think that, for one thing, it's important to have options. And I guess ruxolitinib is a good option for people who have myelofibrosis, but a lot of my patients are actually somewhat resistant to starting the only available drug for the treatment of myelofibrosis because they feel like they're getting to sort of the end of the line. I mean, it must be anxiety provoking for them to feel that way. I really think we owe it to our patients to be able to have alternatives for the treatment of this very difficult disease.
Okay, so let's get into some of these that are you know, hitting our news right now. We'll start with fedratinib for myelofibrosis. Serge, would you tell us about the Jakarta-2 trial?
We mentioned many times that the underlying problem is hyperactivity of the JAK pathway. We also said that there are other mutations, somatic mutations, that contribute to aggressiveness of the disease, and obviously the most of that is in the myelofibrosis. Now, hitting the JAK pathway with the ruxolitinib frontline setting in great majority of the patients would lead to improvement in quality of life and the splenomegaly, but it does not work forever. It works for some time. In studies, on average, is about 3 years. And the longer you follow the patients, you more and more see progressive loss of that benefit. Spleen comes back, symptoms come back, anemia may develop. The patients are in need of something else. And perhaps different JAK inhibitor. Fedratinib is, again, JAK2 inhibitor. It does not touch much on the JAK1. Little bit qualitatively different. It does touch on FLT3 mutation. It has perhaps different activity profile, different little bit toxicity profile as well. Drugs are in the same class, ruxolitinib and fedratinib, but they are different. And fedratinib was studied, in Jakarta-2 study you mentioned, is the study in the patients that were previously exposed to ruxolitinib. And in that setting, the fedratinib was very active. More than half of the patients had significant decrease in the spleen, measured by the 35% by volume, which is usually about half by palpation, and improvement in quality of life. So this tackles what we now consider the primary objective of our efforts, area of unmet need. Life after ruxolitinib is not good, and it's short. People suffer again, no good quality of life. We need another medication. Fedratinib is leading the pack with this promising, very exciting results from the Jakarta-2.
Was there a clinical hold on that drug at one point?
There was, because of venous encephalopathy, that is central nervous system toxicity, which was ascribed to interference of uptake of thiamine with fedratinib from the gut, which may or may not be the case. The 8 cases out of about 800 patients that were treated with fedratinib were scrutinized recently, and it does not appear that this is such a clinical problem that requires us to forget that drug. Quite contrary, the drug has been renewed, and we are looking forward to further testing and perhaps in the near future approval of this drug in either frontline or a second line.
Okay, actually, along those lines, pacritinib. You presented on pacritinib last year or so?
Correct, here at this ASCO meeting, yes. So pacritinib similarly is an active JAK inhibitor. It's an agent whose differentiation sought to be the ability to use it in individuals with marked thrombocytopenia. It's an agent that similarly had gone on a clinical hold, but then subsequently was resolved.
That was cardiac, was the issue?
There was concerns. Again, individuals were allowed to be enrolled even if they had marked thrombocytopenia. So it included a lot of people who had pretty advanced disease. And because of that, there were increased risk of hemorrhage, and there were some cardiac events. Over time, it seemed we learned a few things. One, there probably was adverse patient selection influencing these adverse events. You know, patients were very advanced when they went into the study, so they had a higher risk. Two, there probably were dosing opportunities regarding the safety of the use of the drug, and that was in part borne out by the study that Serge led, the PERSIST-2 study for individuals with a platelet count of under 100,000. So certainly there is an ongoing study seeking to complete accrual soon the PAC-203 study that is delving into a little deeper detail, trying to find the minimally effective dose and to try to inform the entire data package, if you would, on what a pacritinib indication of myelofibrosis could look like.
Okay, so JAK2 inhibitors still being developed here in myelofibrosis.